My name is Emilie Hultin, and I'm a research coordinator at Karolinska Institutet. In this lecture, I will talk about the molecular mechanisms of HPV-induced cancer, and I have divided the lecture in two parts. So the first part, I will talk about cancer genetics and give some examples of oncogenes and tumor suppressor genes. Here we see the normal cervical squamous epithelium, and the dark purple color represents cells that are undergoing mitosis, cell division. So the dark purple color is compact chromatin. In this next picture, we see that these cells has transformed now into cervical carcinoma in situ. And it's more than 2/3 of the epithelium that show severe dysplasia. So why does this happen? Why does normal cells form into cancer cells? We know that there are several different reasons that or causes that can cause cancer. We have chemicals and different kinds of radiation that can cause cancer. Cancer can also be inherited, but some cancers are also caused by viruses. And here we see a human papilloma virus particle in the lower part here. And HPV can actually then cause this transformation into cancer. And what all these causes has in common is that they change the genetic code inside the cells. So for some reason, mutations occur that lead to cancer formation. And for cancer to develop, there's a need for more than just only one mutation. And if we look at the mutation rate within a normal cell, we have a spontaneous mutation rate of approximately 1 error or mutation per 10 gigabases per cell division. And if we look at the human genome, it contains 6 gigabases, 6 billion bases of DNA. So approximately we will have 1 mutation for every second cell division. And for a cancer for a normal cell to convert into a cancer cell, usually it's necessary with approximately 6 or 7 mutations for this transformation to occur. But it cannot be any mutation. It needs to be mutations that either enhance the cell proliferation, because if you have mutations that increase the cell proliferation, it will create an expanded target of cells that grow much faster than the rest. And this in turn will lead to, will then give rise to more mutations, as if we have more cells that undergo cell division, it's more likely that a second mutation occur. So the more cells that divide, the more likely it is for a second mutation to occur. It can also be mutations that affect the stability of the genome. So instead of just increasing the cell division, it can actually increase the mutation rate instead. And that could also then give rise to more and more mutations. So eventually we might have 6, 7 mutations that eventually give rise to cancer. And looking at the cells within the tumor is very heterogeneous, which means that the cells are not the same anymore. The tumor starts with 1 cell, but then during the cell divisions, the cells, they will gain more and more mutations. So within the tumor, there will be a lot of different mutations within different cells. So if we're looking at the cell cycle, there is a lot of different genes that control the cell cycle. We have genes that control the proliferation of the cell, the differentiation of cells, and the survival of the cells. And these genes that control cell cycle and survival, these genes are called protooncogenes. These are the genes that promote growth of the cell. And we have tumor suppressor genes. These are genes that inhibit the growth of the cell, stop the proliferation, or lead to cell death. So here we have some examples of different proto-oncogenes that promote the cell growth. We have growth factors. One example is the EGF transcription factor. And we have growth factor receptors. We have signal transduction proteins that send the signals from the cell membrane into the nucleus. We have also transcription factors and cell cycle proteins like the cyclins. And then we have also apoptosis inhibitors that inhibit cell death. The tumor suppressor genes, on the contrary, they inhibit the growth. So tumor suppressor genes, they promote DNA repair and apoptosis and other cell deaths like senescence. They can also, on the contrary, they stop the cell proliferation, and they also stop immortalization and anchorage-free growth of the cells. And they stop cells from being invasive. So the balance between the proto-oncogenes and tumor suppressor genes, that is the key to life. That is what controls the cell cycle. And when cells turns into cancer cells, this balance between proto-oncogenes and tumor suppressor genes is altered. So what happens is that the cancer cells, they become masters of their own destiny. So they can produce the growth factors themselves, so they are not dependent on other surrounding cells. They can send signals to stromal cells to provide them with different growth factors. They can also increase level of receptors of the cell surface so that they are more sensitive to different growth factors, or modulate the receptor structures to be more sensitive. They can mutate or activate downstream pathways and disrupt the negative feedback mechanisms of the cell cycle. So instead of having a normal or like a balance with the growth and death of the cells, cells starts to grow too fast and too much. So how can then a proto-oncogene become an oncogene? Yeah, there can be different kinds of mutations or on the DNA level. So if we're looking at the first part here, we can have a mutation that activates the proto-oncogene. It can be a mutation within the gene of the DNA that actually produces then a hyperactive protein. This protein can still be in normal amounts. And one example of this this kind of activating mutation is usually seen in the Rus protein, the Rus gene. And Rus is an enzyme important in the cell cycle, and it's mutated in 30% of all tumors, and especially in colon and pancreas cancer. Another way for a proto-oncogene to become an oncogene is that the gene instead is amplified. So instead of having a mutation, the whole gene is amplified. So instead of being just one copy of the gene, we can have several copies of the same gene. And this leads to a normal protein. However, it will be in too high amounts. So we will have an overproduction of this protein instead. And one example of this is the ERBB gene in breast cancer. This is quite common in breast cancer. Another way for a proto-oncogene to become an oncogene is that there are some chromosomal rearrangements. In the first step here, we can see that here we have some rearrangements that this green DNA part comes from another chromosome or another part of the same chromosome. And now it has fused together with this proto-oncogene. And this leads to, this is like a regulatory sequence. and it will lead to an overproduction of the proto-oncogene. So here we also have a normal protein, however, in higher amounts than normal. And this is quite common in Burkitt's lymphoma, where this MYC protein is overproduced. There could also be another way of chromosomal rearrangement is where we have a fusion gene, where this orange gene from another chromosome has fused together with the red gene and form a fusion protein. And this fusion protein is either highly expressed or hyperactive. One example is in chronic myeloid leukemia, where the BCR gene is fused together with the ABL gene and form a hyperactive protein. So here is an example then of the enzyme RUS, which is involved in the cell cycle stimulating pathway. So here what we see here is that we have the cell membrane, we have a receptor and a growth factor that binds to the receptor. And what happens then is that this bridging protein will bind to the RUS enzyme. And this RUS enzyme then becomes active and can then activate the MAP kinase pathway. And this kinase pathway will go into the nucleus of the cell and activate transcription of different cell cycle proteins. So the cell cycle will progress. The thing with this RUS enzyme is also that when this MAP kinase pathway is activated, there is a negative feedback loop where a phosphate group from the active RUS is released so that the RUS is then inactive again. So it has this negative feedback loop. But when we have a mutated RUS that is common then in different tumors, this negative feedback loop doesn't work anymore. So with a mutated RUS enzyme will always be active, and that leads to cell cycle progression, that we will have activation of the kinase pathway, continuous activation of the pathway, and the cell cycle progression. EtoF is a transcription factor, and this one is an oncogene when it is mutated. The transcription of S-phase genes are controlled by the EtoF transcription factor. When EtoF is bound to the DNA of, or like to the genes, different cyclin genes, they will be transcribed, and the cell cycle will continue from the restriction point into the synthesis phase. And here we have a tumor suppressor gene, the retinoblastoma gene. This RB gene is a master break of the cell cycle. So it's the opposite to this transcription factor that I was EtoF. So RB protein stops the cell cycle it stops the cell cycle before the synthesis phase. And this RB protein can either be phosphorylated or not. What happens with these? How does this work then? So retinoblastoma protein and the transcription factor EtoF works together. So the retinoblastoma protein it can bind to the transcription factor. So when the retinoblastoma protein binds to the transcription factor, the genes that are needed for the synthesis phase in the cell cycle are not transcribed. However, when there are some growth factors that bind to the cell surface, it activates this RUS pathway and activates the cyclins to be transcribed. And this also releases phosphate groups. So after that, this RB protein is phosphorylated and cannot bind anymore to the transcription factor EtoF. This, in turn, leads to gene transcription of the different enzymes that are needed for the S phase, so the cell cycle will progress. So that's why the RB gene is a master break of the cell cycle, and the EtoF transcription factor is a proto-oncogene because it activates the cell cycle. Here we see another tumor suppressor gene that is very famous. It's the p53 protein. This p53 protein is the guardian of the genome because it can stop the cell cycle and it can give rise to apoptosis and activate those apoptotic pathways and DNA repair pathways. So here we see a big picture of different examples of p53 action within the cell. So looking at this, there are like 3 different examples here. So in the top left corner, we have a happy cell with a normal p53. When we have some DNA damage of the cell, the p53 is activated and it will bind to DNA. And then it will activate some transcription of different genes. The transcription of P21, which will turn the cell into G1 arrest. And it will also activate the GAD45 gene, which is a DNA repair gene. And if the repair of the DNA is successful, the cells will continue to divide and become normal cells. However, if the DNA repair fails, the p53 will activate the apoptosis pathway and the cell will then turn into apoptosis and die. So that was the happy normal cell. However, if we have cells with mutated p53 or the p53 function is lost, if we have DNA damage in certain cell, these p53 dependent genes will not be activated, so the cell cycle will not be will not turn into arrest and will not be stopped. The DNA will not be repaired and the cell will not die. So we will have a mutant cell that can continue to grow and expand, and eventually it can form this malignant tumor of mutated cells. And more recently, another function of the p53 was discovered, and that is that the p53, when there are some DNA damage within a cell, the p53 can also activate a microRNA. And this microRNA can inhibit translation of growth-promoting genes and turn the cell into quiescence or senescence. The microRNA can also inhibit translation of some anti-apoptosis genes, and that leads to apoptosis of the cell. So that's another pathway of the p53. And looking at the degree or like the number of, or like what to say, yeah, the number of p53 mutations in different human cancers, we can see that a lot of cancers have mutated p53 genes. It's very common in different human cancers. Here we see a table of different cancers, and on top we have ovary and rectum cancers, and they have approximately 51% and 47% of ovary and rectum cancers have mutated p53 genes. However, if we then take a look at the bottom of this table here, we see that cervical cancer in the cervix uteri, cervical cancer, it's only 6% of these cervical cancers that have mutated p53 mutations. So I will come back to this in the next part of the lecture and talk about why this is the case. But the take-home message from this first part is that cancer development, it occurs through genetic changes and clonal expansion of the cells. And the cell cycle balance is disturbed by activated and inactivated genes. And an oncogene is a gene that promotes cell division or protect against apoptosis. And an oncogene is activated in the tumors. On the contrary, a tumor suppressor gene inhibits cell division or promotes apoptosis, and tumor suppressor genes are inactivated,